Those are fun.
All right, so I think we'll get going. Let me hit the record button, lest I forget. Click. All right, I'm just going to move this. Recording. All right. So hopefully, so before we get into today's lecture, I sent out everybody, I'm just going to close this out, see if that works. I sent out an announcement to everybody. There's this really cool thing called Immune Quest. It's a game, so all the gamers out there, take note. That covers actually the rest of what we're talking about in this block, in our innate immunity block. Complement, macrophages, things like toll-like receptors that we'll talk about starting next week. This game involves all of these components where you get to try to save a host from an invader. Think of it like learn by doing. It's a really great way to study before our first test. That's actually really fun. I encourage everybody to at least check it out. All of the instructions to get onto Immune Quest and to get into my course that I've set up is in that announcement. And just as added incentive, because I know, there's going to be prizes. So come Thursday, February 16th, so it's after the test is done, but at 9 a.m., I will open up my version of Immune Quest and find the top 2 score getters. And what do those people get? Oh, somebody's got something in the chat. Prizes, yes. And I just want to show you, because I'm really bad with the, here we go. Let's see if this works. Oh, gosh darn it. Come on, boo-boo. There we go. Prizes. Yes, they are at my house right now. And gosh darn, they are cute. First prize, that's an antibody binding to an antigen. That's what we're going to be talking about starting Thursday, February 16th. Second prize, second highest score. What is this little guy? Well, it's a white cell. Ooh, could it be a leukocyte? Absolutely. So the 2 highest scores will be awarded these prizes at the beginning of class on February the 16th. So again, it's a great way to learn as well and to have some fun with it. And it's produced by the National Science Foundation, so it's not some kind of weird ass stuff. All right, so let's get going then. If you haven't figured it out already, and as I've kind of subtly hinted to, I like to start each class by kind of doing a little bit of reviewing or going back to the highlight reels from the previous class. And of course, our last class was barriers. They were everywhere, as Buzz says to Woody. So here's the thing, and I think kind of weird at times. You probably haven't noticed that. But when we start to talk about our frontline barriers, this is our first stage of innate immunity. I want to show you what I think any microbial pathogen, anything trying to invade our bodies, views us at. So here you go. Ta-da! That's how I think microbes view us. And you're looking at somebody who's completely decked out in personal protection devices, right? The gloves, the shoes, the aprons, the whole kit and caboodle. Well, that is us. We've got a ton of frontline barriers. And you can put them into three major categories. There they are. We have mechanical barriers that we talked about in last class. We've got microbiological barriers. We've got chemical barriers. And we've got multitudes of things going on in each of these barrier systems to help us stop any pathogen from getting in. Because it's very simple. If the pathogen can't get in, it can't cause any disease or harm to us. Which leads us then, as we're reviewing, to my first top hat question. And steeping some tea, that's what I'm drinking right here. So here's the question. Competitive exclusion, we talked about that last class, is associated with what general barrier type? So here are your choices. Is competitive exclusion associated with immunological barriers, mechanical barriers, microbiological barriers, or chemical barriers? What is competitive exclusion associated with which barrier type? So I know it's a little harder to now introduce yourself to people around you because probably everybody's sitting at home, but feel free to look over and pull up the slides from last week. See what you can find. And remember, competitive exclusion, when you're competing, it's a living entity competing against another living entity. That is my hint for today as well. What's competitive exclusion associated with? What type of barrier system? And again, remember, competitive, we compete against one another. We are two living sentient beings. Well, competitive exclusion is between two living sentient beings. You got 30 seconds left. And remember, hitting any of the 3 or 4 choices nets you a point regardless. The second point comes from getting it right. What's competitive exclusion associated with? What barrier type? All right, in 3, 2, 1. All right, let's see what we've got. The vast majority of you said microbiological barriers. And if you think about it, microbiological, well, that's microbes. They are living, right? They're really small, but they're living beings. And if you pick microbiological, that is absolutely correct. Competitive exclusion is when the normal flora we have already on our body excludes any pathogen from coming in because they've either already bound to receptors in a certain area or they've used up the nutrients. And so those new incoming microbes, the bad ones, the pathogens, the foreign invaders, don't have the nutrients or the receptors to stick around and cause disease. So when we talked about our barriers, and that immunological one, I threw that in, that was just a total fake red herring. We have got mechanical, that involves what we talked about, things like tight junctions between cells. The cilia that's on our cells that are batting things back out, especially in our respiratory tract. And then mucus, this goopy stuff that actually traps foreign microbes. We got the microbiological barriers where there's competitive exclusion, the fight for food, and some microbes in our bodies already, our normal flora, make antimicrobial substances like bactericidins, little bacterial antimicrobial peptides. And then the chemical barriers, we talked about a bunch of those, things like sebum, the oil on our skin, pulmonary surfactant in our lungs, lysozyme and secretory phospholipase A. These are both enzymes. So think kind of big proteins. And then those little teeny weeny, but really cool antimicrobial peptides. Just to remind you, the last thing I talked about, these antimicrobial peptides, and there's a couple of really cool GIFs as you're watching bacteria in this case kind of start to shudder and then die down here. You're watching these AMPs, these small catatonic amphipathic molecules insert into a membrane. And that membrane, unfortunately, happens to be on a bacteria. And there's a lot of research going on now looking at using AMPs as a new form of antimicrobial agents. And I didn't get around to this last couple slides in our barriers PowerPoint slides, but I want to let you know that I have put up a video lecture. So if it mentions anything about COVID, because that's when it was recorded, don't panic. But it's in the module on mechanical, microbiological, and chemical barriers. And it's a wrap up because in any one of our spaces, whether it's our skin or our lungs or our gut, it's not just one barrier working, it's multiple barriers working. It's multiple mechanical, microbiological, and chemical barriers going. So that, and it's about an 8-minute video, that kind of just brings everything back together. So if you want to watch that, please feel free. But as in anything in life, nothing is perfect. There's sometimes a breach. Right, as you see here, this person hopping the fence. Well, that fence was our barriers. What happens, because we've all gotten sick sometime in our life, we've all had that breach. What happens when we have a breach? What's our next line of defense? How are we going to stop this from happening? Or how are we going to stop this, in this case, if it's a pathogenic microbe, from causing disease? So if you kind of look at this flow chart here, where we've got the pathogen, our containment barrier has failed, we now have the potential for infection. But right away, we throw up, not throw up like vomit, but throw up and out to the pathogen another innate defense mechanism. And this is a defense mechanism initially discovered by Jules Bordet. As you see here, he also discovered the pathogen Bordetella pertussis that causes whooping cough. He won the Nobel Prize in 1919 when he discovered something called complement, or the complement system. It's what we call part of our immediate innate immunity. In other words, it's on all the time, just like our barriers are. So what is this complement system? Well, let's first talk about the crazy name, complement. Well, it was named the complement system because it complements, it enhances our other immune mechanisms, things like phagocytosis, our immune cells gobbling up, eating a pathogen, or antibodies that we'll talk about starting in February. The complement system works with these types of pathways and mechanisms in order to have us have the best or most robust immune response. Now, the complement system starts in the liver. It's over 30 proteins, okay, that are produced in the liver and then put into circulation. And when I mean circulation, they are always in our blood, they're in our lymph, and they're in any extracellular tissues in our body. So we're kind of bathed in all of these proteins, right? They're always with us, turned on all the time, made in the liver. So the important things to know about this complement system. Important thing number one, most of those 30 proteins are proteolytic enzymes. Specifically, they're serine proteases. And for those of you who are a little, didn't like biochemistry, proteases, they're going to break down proteins, right? They're going to break them into pieces, specifically with serine proteases at a serine site. Many of these proteolytic enzymes, these complement proteins that are made in the liver, they are produced and made in what we call an inactive form. In other words, they can't chew up any proteins right away. They're non-functional. They're what we call either a proenzyme, or a word you're going to see a lot are zymogens. Zymogens are proteins that are not yet activated. But obviously, somewhere on the way, they're going to get activated. What turns on these proteins is infection. That's the trigger. That's, in a sense, the finger that pushes a bunch of dominoes over. And I'm saying the word dominoes, like that game where you watch dominoes fly, because these complement works in a chain reaction where one molecule is going to have an effect on the next molecule. Well, let me show you in picture form. So let's say this down here, this blue circle is our first complement protein. It's not functional. It's a proenzyme or a zymogen. But upon infection, there's going to be a change to that enzyme, and it's going to become activated. In this case, part of it is removed to indicate an active site. Now we have an active molecule, an active enzyme. And that enzyme is going to cleave the next enzyme in the sequence. That's the red guy here. Again, a proenzyme, a zymogen, inactive. Its activity only comes into effect when this active enzyme works on it. In this case, it's going to protease cleave this portion out. Now we got another active enzyme. And we play this game going from one enzyme to the next, all through the complement system. It really is a giant set of tumbling dominoes, right? Where that's the first enzyme that becomes activated. That one's going to then activate the next one, which was inactive normally. And then that's going to go on to the next one and the next one and the next one. Complement is really cool because not only is it a cascade effect, but it's an amplified effect, as you'll see, that it's not just one domino, it's going to be massive dominoes falling at the same time. And that's going to lead to elimination of the pathogen. So, believe it or not, we don't just have one complement pathway, one activation way of getting these proteins to turn on. We actually have three complement pathways. The alternative pathway, which is the first pathway to act when a pathogen comes into our body. Something called the lectin pathway, that's the second one that's activated. And then we have the classical pathway, the last one activated. All of these three will eventually get turned on during an infection. But what turns on each of these pathways? And the thing is that each of these pathways is turned on in a different way. For example, this alternative pathway, which we're going to start talking about in a minute, it's all based on the local environment that turns on that first complement protein. That's the finger that pushes the first domino down. When we're talking about the lectin pathway, now we're talking about specific lectins binding to a pathogen surface. That's the first finger to push the dominoes down. And the classic pathway, well, that's things like antibodies or C-reactive proteins. And don't panic, we'll talk about all these. If you don't know what they are, no problem. But I want you to know from this, A, that we have three pathways and that each is activated, turned on in a different manner. So let's start by talking about the first pathway to act, our alternative complement pathway. So this pathway, not only is it the first one to be turned on, it's the most ancient of the pathways, right? Although it was the second one to be discovered by scientists, it is actually the oldest pathway we have. And as I said before, it's turned on, or it's immediately there right after a pathogen invades us when we have a breach. It's our first response. And the most important protein for this alternative complement pathway is a protein made in the liver called C3. C3, yeah, so they're highly technical names, complement component 3. Now, let me show you a kind of cartoony picture of complement component 3. Because remember, this is a protein, right? This is a serine protease, but it's inactive. So it's a complex molecule, right? And as you see it here in this kind of cartoon, we know that it's inactive. It's actually C3, the highest concentration of a complement component in our blood. And the most important thing to know is that buried in this structure of C3 is a thioester bond. Now, this thioester bond is buried in the molecule, so it is not exposed to other components. It's hidden, it's masked. But the really cool thing is that this C3 that's in our blood, most of the time it looks like this, but C3 can very slowly and very infrequently go through a conformational change, a structural change. It changes its shape. Doesn't happen very often. It happens kind of slowly. It's not like every molecule, every C3 molecule in our body is all of a sudden changing. But when a C3 molecule changes its shape, all of a sudden now this hidden thioester bond becomes reactive. It's exposed. And when it's exposed, then it can be hydrolyzed by water. And that's really important because when the water hydrolyzes that thioester bond, all of a sudden now we have an active molecule. Let me see if I can do this. The slides on Top Hat aren't updating. I don't know how to fix that. Is everybody having that problem? Anybody want to be brave and just hit the mic and say yes or no?
I'm not having that problem.
Okay.
Yeah, mine's.
Okay. Lizzie, you might, sometimes you just have to rejoin the class to fix it. Did you hear that, Lizzie? Sometimes you have to rejoin the class to fix it. Or press the catch-up button. Sorry about that. Welcome to the world of technical difficulties. But when this thioester bond, shape change, now thioester bond is exposed, water gets to it, we have another change in conformation to produce this molecule, and you can call it one of two names, C3H2O, kind of long. I like to call this molecule IC3, because this is C3, and we have now activated it. Please don't think the I here stands for inactive. It does not. Now this molecule is active. So what does this molecule do? Well, here we go with the steps. So this first step is what I just talked about. And as you're going through complement, this is a great way to study, is to go through the steps. All right. So step one, there's our C3. It's in our blood. There's plasma there. Let's say there's a pathogen in our body. There's C3 from our blood, right? It's going to be a conformational change. That thioester bond gets shown, and it may be just one molecule. All it needs, water is going to come in, hydrolyze that thioester bond to now make this active molecule called IC3. And now comes the second complement protein. Complement protein called factor B. Now, factor B, believe it or not, you're going to see this trend here, is another inactive protease. It's a zymogen, can't work. Its conformation will not allow it to work. But what it can do at this point is bind to IC3. And that binding effect is going to change then factor B. It's going to allow factor B to expose a proteolytic cleavage site. In other words, the site says, here, cut here. That happens only when factor B binds to this IC3. Factor B can't bind to C3, only IC3. And so now the question is, well, what's going to cleave this proteolytic site? That's step 3. One of the few complement factors that is actually active right from the get-go is factor D. There you see factor D. Again, another protease. So it's great for this cleavage, and it's already active. Nothing has to be done. It's ready to go. It's going to come along and cleave factor B into 2 parts. Factor BA, and this is going to be like a bad alphabet soup, so be prepared. Factor BA, that's going to kind of float away in the bloodstream and eventually be degraded, and factor BB. And factor BB remains bound to IC3. And so now we have this new molecule right here that we can call, again, bad alphabet soup. I did not, name the complement components, so don't blame me. This one's called IC3BB. And believe it or not, this component, soluble, it's floating around your bloodstream, is actually now an active enzyme. So there's a bigger picture of IC3BB, right? Where that thioester bond that was deep in here, there was a conformational change, water came along, caused another conformational change to make IC3, and then factor B bound, was cleaved, and now we've got BB. This is what we call a fluid. In other words, it's soluble, it's still floating around in our fluids, a C3 convertase. So it's an enzyme. There's the ACE at the end. A C3 convertase, what's it going to do? It's going to convert C3. In other words, it's going to cleave C3 molecules that are floating around, normal C3 molecules in our blood. And it's going to cleave it into 2 component parts, C3A and C3B. Oh, let's see. So is IC3? Yes, so IC3 is active, but it doesn't have any proteolytic activity per se. Its activity is allowing factor B to bind. Factor B. So somebody asked, so is IC3 and IC3BB both active? Answer is yes, but the activity is slightly different. IC3, its big activity is just the change in conformation to allow factor B to bind. IC3BB is actually now an active serine protease. That's what you're seeing here. This pair of scissors is actually your fluid C3 convertase coming in. Ooh, let's see. Oh, you're welcome. So that scissors, think of that as your fluid C3 convertase. This molecule here was just your regular C3, right? Had that hidden thioester bond, but because this convertase is going to actually cut the molecule, it's going to now expose that thioester bond. And so the C3 convertase, this fluid C3 convertase, is going to give us 2 products. C3A, this small portion that's going to float off, and it's actually, we're going to talk about it much more on Thursday. It's a mediator of inflammation. C3B, the bigger component, is going to now have that thioester bond exposed. That's really important. This wasn't a conformational change now. This was because of this fluid C3 convertase. Is fluid C3 convertase the same as IC? Yes, they are exactly the same. IC3BB is the fluid C3 convertase. Because I hate to tell you this, a spoiler alert, there's going to be another C3 convertase we'll talk about in a minute. Put the I in front, that means it's fluid, it's soluble. So here we have this now cleaved C3b. And when it was cleaved, now it's showing this thioester bond. It's exposed. Now it can undergo nucleophilic attack. And it can be attacked by one of two things. Water. If water attacks this thioester bond now, what we're going to end up with is an inactive soluble C3b, right? Not going to be functional. All it's going to do is float around the blood and eventually going to be degraded. So this is nothing great. But if this C3b with its thioester bond that's exposed now is close to a pathogen surface, what it can do is have this nucleophilic attack caused by an amino acid or a hydroxyl group that's close by. That means amino acid, well, any protein on the surface of this pathogen, or hydroxyl group, any carbohydrate. If this is the first area of attack, what's going to happen is that a covalent bond, emphasis covalent, not easily removed, is going to allow this C3b to bind to the pathogen. This may not seem very important, but it is the start of the death of that pathogen. In this case, I'm showing you a bacteria. When any microbe gets C3b covalently attached to it, right, that marks that pathogen for destruction. And we call that destruction, or we call this marking, sticking, think about it like sticking C3b into that pathogen, gluing it in. That's what we call complement fixation. Complement, because C3b is part of the complement pathway. Fixation, this is because of the covalent bond. This is not easy for a pathogen to get rid of. It can't just shake it off. So let's kind of go back and rehash. There's what we have just talked about, all right? Going from this C3 that's floating around in our bloodstream, for example, conformational changes, it slowly over time exposes that thioester bond itself. There's no cutting. This is a natural occurrence. We get IC3. That molecule is now active and can bind factor B. Factor B binds, changes conformation, shows that proteolytic cleavage site, which enables factor D, so far the only complement factor we've talked about that is already active, to come in and cleave factor B. That leaves us with this guy, IC3BB, our fluid C3 convertase. Now, this floating enzyme, any other C3s it encounters in the blood, remember C3 is big in the blood, it can cleave that C3. It can cut it. So IC3 bond is not exposed like C3b, but it can still use that to bind factor B. Correct. So the question was, IC3 thioester bond was exposed in a completely different manner to go from C3 to IC3. That was a natural occurrence, just a conformational change that happened slowly. But over here, when we're talking IC3BB and that enzyme now can cut a regular C3 to expose the thioester bond in a completely different way. And that exposure of the thioester bond, think of it like being right smack in the middle here, is what allows now that thioester bond to be exposed, hopefully close to the surface of a pathogen, which allows covalent binding of this IC3B. That's from this molecule. That's the enzyme, cleaves this, opens up the thioester bond now, and if this is in close proximity, it plonks itself down covalently on that surface. If not, it will be hit by water and becomes an inactive molecule. You get an inactive C3b, and it floats away. And I said this complement fixation spells the death of the pathogen. Oh, I hate when that happens. Because this is only the start. Remember I said complement is a cascade effect. You can see the cascade, but it's also an amplified process. Let's see. What makes the C3 in our bodies? It's made in the liver. Our cells have the gene for C3. It's like any other gene in our body in the genome. So it's one of the genes. I don't even know what chromosome it's on, but we make a lot of it in the liver, and then it gets expelled into their blood and our tissue or our fluids. Once this little C3b gets plonked down, covalently attached to the surface of the pathogen, guess what? Factor B can come in, that's in our blood as well, and bind to this C3b. Now, this is all happening at the surface of the pathogen. Factor B comes in, changes conformation once it binds, proteolytic cleavage site opens up, factor D comes in. Factor D, an active serine protease, cleaves factor B. What we end up with, once again, is BA floating away, BB still stuck to C3B. Hey, wait, we've got a C3B, BB. This is our pathogen-bound alternative convertase. So there's two C3 convertases. Our fluid one, that's up here, right, floating around all the time, IC3BB, and then our bound version, C3BBB. Only difference, the I here indicates that this is our fluid. This is an active enzyme as well, an active serine protease. And what it can do is the same thing. Any C3 in our blood that comes into contact with this enzyme can be cleaved into two parts. C3A floats away. We'll talk about it, causes inflammation. C3b, and guess what happens to C3b, especially here where it's being cleaved so close to the pathogen surface? That cleavage opens up that thioester bond, looking for a nucleophilic attack, and because it's so close to the pathogen surface, easily lots of amino acids, hydroxyl groups on the pathogen surface, it gets plonked onto the pathogen surface as well. And this keeps happening. So I like to think of it this way. That first complement fixation, that first C3b down on a pathogen surface, that's like this sticker on this girl's forehead. I should have put like C3b on there. That marked her or the pathogen for death. But that also allows now multiple C3bs to now get put on with this amplification effect. So by the time that poor pathogen, sits around for a little while, it's going to be covered, covalently attached C3b. So while this marks it for death, we stick a whole bunch of C3bs on its surface. Complement fixation. That means that pathogen in this case is not going to stand a chance in our system. And we'll talk a lot more about how we're going to kill it in a little bit. Now, a couple of slight problems with our alternative complement pathway. First problem, this C3BB convertase that's stuck on the pathogen surface, right? Covalently bound. Unfortunately, though, it's not very stable on the pathogen surface. Easily prone to degradation by other proteases in the system. Not those that relate to complement, but just other proteases we have. Well, that's not good. We don't want this degraded if it's on the pathogen surface. This is what's supposed to mark the pathogen for death. We want to stay there. And so we have to have a positive regulator. That positive regulator of this convertase, right? This is our pathogen-bound C3b-BB convertase. Our positive regulator is a molecule called propyrin, otherwise known as, this is really easy, factor P. Factor P for positive regulation, binds to this component, binds to this enzyme, and allows it to be stable. It still allows it to be active. As you can see here, it's binding now. This is nice and stable, not going to be degraded. C3 comes in from our bloodstream. It's going to be cleaved. C3A floats away. C3b, if it's close to the pathogen surface, it's going to be covalently bound. And we're going to get this massive amplification effect. So we've got a positive regulator. We need factor P in order for our pathogen-bound C3 convertase to remain, not degraded. But we also have to have some negative regulators of our C3b-BB. Because here's the little problem. There's the fly in the ointment. That C3b, remember I said once it's cleaved, several things can happen. If it's cleaved and water attacks it first, we get an inactive C3b. Oh, well, it just goes off and gets degraded. If it's close, however, to amino acid or a hydroxyl group on proteins or carbohydrates, and we were hoping those were proteins and carbohydrates on the pathogen surface, you undergo a nucleophilic attack and you have covalent binding. Well, the problem is that our cells, our host cells, have amino acids and hydroxyl groups on their surface as well. Uh-oh. We don't want the C3b, there it was, opened up, right? There's the thioester bond. If it's water, inactive molecule. If it's close to a surface where there's an amino acid or a hydroxyl group, we get a covalent bond. Now, the hope was that that would be the pathogen surface, but it could be our surface. Hold on. If you hear my cat, just don't pay any attention. I thought he was going to sleep through the day, but oh well. This could be the host cell, our cell. The last thing we want is to put complement on our cell, marking it for death. The whole idea of our immune system is to kill the pathogen, don't hurt us. This, if it's on our cell, is a big problem. So we have to have some negative regulation. We have to have some ways, because this could happen. We have to have some ways of preventing this. So like I said, how do you stop host cells from inappropriate complement fixation? In other words, once again, how do you discriminate us, our cells, from non-self, from foreign entities? And so we have a bunch of negative regulators of this C3b, BB, C3 convertase that's stuck on the surface of a cell. We hope that surface is a pathogen and not our cells. But if we do, for example, have this enzyme now stuck on the surface of our cell, because this can lead to complement fixation, we have a factor in our blood called factor H. And factor H likes to bind to sialic acid residues. Now, here's the great thing. Sialic acid residues are typically on eukaryotic host cells, like our cells in large numbers, but sialic acid residues not very often seen on things like fungal yeasts or any bacteria. So right now we have a factor H, that is very specific for binding mostly to eukaryotic human cells. And when factor H comes in and binds to sialic acid residues on the surface of that cell, if there's a C3 convertase, what it can do is knock off, displace the BB portion of that convertase. That means right now that this convertase is inactive. It doesn't have part of that convertase, the BB part. Now, just to make sure that this molecule here will never take another BB, factor H also is a cofactor, is required for another protease associated with our negative regulation called factor I. And factor I can come in once factor H is bound. It's displaced the BB. Factor I is a protease, and that's got looking like a Pac-Man there. What's it going to cleave? Well, when factor H is there, brings it on in, it's going to cleave and release factor H, so factor H can be used again, but it's also going to cleave this C3b. And it's going to cleave it such that what we end up with on our cells is an inactive IC3b. Please don't get confused. I know it's crazy how they've done it, but this I, when you talk about IC3b, and this is the important part, the little b, that means it was only half of the C3. This is an inactive molecule now. It cannot bind to BB. It can't do anything. It's a dud molecule on the surface of our cell. We don't get complement fixed. Factors H and I do that. We also, believe it or not, have actually receptors on a lot of our cells that can also act as negative regulators. For example, we have a molecule called DAF, decay accelerating factor, and you can see the cartoon of it here. If we end up with a C3b, BB, that's our C3 convertase on our cell, DAF can come along. And if it's on the same cell, remember, we have fluid motion of our cell membranes, can come over and it actually can bind and displace factor B, factor BB. It bumps it off. It bumps it off so that this molecule now is inactive. It's a daft C3b conglomerate that factor B cannot now bind. So we've taken out a potential complement fixation problem with DAF. We also have another regular membrane-bound surface protein called membrane cofactor protein, MCP. It is another negative regulator. It's on our cells, right? And what it can do, MCP, it can also bind to this C3 convertase. And in this case, not only does it displace factor BB, knocks it off, but it also allows for a conformational change in C3b to allow that factor I talked about a few minutes ago to also come in and cleave this C3b. And when it cleaves the C3b, we once again get an inactive form. So we're taking this active C3 convertase that has accidentally been put onto our cells, that we don't want the C3 convertase on our cells. That's going to lead to complement fixation. We're going to disable it by either factor H and I working together, by DAF proteins on our cells, or by MCP and factor I. All of those are negative regulators. And we have to do that for our alternative complement because it's not specific. Is C3b still active when only associated with DAF? No, it is not. It cannot. So the question was, is C3b still active when associated with DAF? I can go backwards there. Boop. It is not active at this point because there's a conformational change that occurs and factor B cannot rebind. So C3b by itself is useless. It has to have that factor B put on. Oops. So we have to have all of these negative regulators because this alternative pathway, it's not specific for any microbial signal at all, right? The initial start to all of this is simply a natural, very slow, very infrequent conformational change. That sets everything off. And so while we have C3, factor B, and factor D, and factor P, all required to turn on the system of the alternative complement pathway, we also have all of these regulators of the pathway, like factor H, I, DAF, and MCP, to make sure that if we accidentally get a C3 convertase stuck on our cells, that we can turn it off, enable it, inactivate it before it does any damage to our cells. So that's the one thing with the alternative pathway. It's not specific to the pathogen at all. That's why we have built-in regulators. So let's just review a little. I know this is it's like a massive bad alphabet soup. I apologize. Like I said, I don't shoot the messenger, but here we go. There's our C3. This is the one in the bloodstream, right? That first one, there's a thioester bond hidden in there somewhere, hidden, masked, that over time will eventually become exposed. Very infrequently, over time, very slowly. But when that happens, we get IC3 being made, right? Water can come in, and in this case, the water does not inactivate. This is full C3. What it does is it changes the conformation enough to allow this molecule to be produced. And its claim to fame is the fact now that it's gone through conformational changes to allow it to bind to factor B, right? All this is under fluid conditions. Factor B comes in, it binds, it changes its conformation, opens up a proteolytic cleavage site. Factor D, our token active protease comes in, cleaves factor B, and what we end up with is something called IC3BB. And the question is, what's IC3B and what does it do? Well, that's our next top hat question. Excuse me. So the question is, what is IC3BB and what does this enzyme do? And you only get 3 choices here, so you got, a 33% chance. IC3BB is a soluble zymogen that cleaves factor D. IC3BB is a fluid C3 convertase that cleaves C3. Or is IC3BB a surface-bound protease that cleaves factor, P. Let's call it P. So what is IC3BB? It's an enzyme. What does it do? So feel free to look back on your PowerPoint slides and all of that. What is IC3BB and what does it do? 30 seconds left to put in your answer. Remember, for hitting any of those three, you get a point. Excuse me, if you get the right one, then you get a second point. What is IC3BB and what does it do? In 3, 2, 1, let's see what we got. Okay, so the vast majority of you think it's that IC3BB is a fluid C3 convertase that cleaves C3. And if that's the answer you picked, you're absolutely correct. So IC3BB is the one that is floating around in the bloodstream. Like I said a moment ago, and we'll see it in the next slide, that was based on it becoming IC3 just because it changed conformation, showed this thioester bond that could then be cleaved by water or hydrolyzed by water to allow BB, factor B, to bind. It's not a zymogen, it's an active molecule, right? It is a ready to go protease that can cleave not factor D. Factor D is an active serine proteases, but it's an active molecule that can cleave C3 in our blood. So there you see, there we going through the process. This guy right down here, active C3 convertase in fluid form. That in the fluid form can now, once it bumps into a C3 molecule in the bloodstream, can cleave that C3 into two parts. That's that little kind of line, it's cut line, right? That's not what's happening here initially. This molecule is going to be able to cut any C3s. We're going to get two molecules, C3A, we won't worry about that yet. C3B, that's the one that can get plonked down because now we have that thioester bond showing. And if it's in the presence of an amino acid or a hydroxyl group, we can get that covalent bond formation. That's complement fixation. Very difficult for a microbe now to remove this, right? Once this is on the surface, now factor B can bind. Same thing as before, factor B was inactive. Once it binds, changes conformation, shows a proteolytic cleavage site. In comes factor D. Factor D cleaves factor B. Remember, factor D is the only active serine protease we've talked about so far. Factor B, when it's cleaved, factor BB, the larger part, remains bound to factor C3B on its surface. C3B, BB, what is this? What can it do? Don't panic. No, it's not another top hep question. What this is our bound version of our C3 convertase. What it can do on the surface of a cell is the same thing. It can cleave C3 that's floating around in the extracellular fluids. And when it cleaves, we go back through this process again. This process becomes amplified over and over. Right, there it is, alternative C3 convertase, cleaving C3. We have to have, don't forget, our little positive regulator, right? This comes in right in here to keep this molecule from being degraded on the surface. Factor P, positive regulator. And we also have to have all of the negative regulators I talked about, because this event, putting this on the surface, isn't pathogen specific. It's just looking for an amino acid on the surface, a hydroxyl group on the surface. And holy heck, our cells have a bunch of those on the surface too. So we have to have that negative regulation because we don't want to accidentally complement fix our cells because that leads to death. Okay, now you're probably like, oh my gosh. Yeah, okay. So I apologize because it's not going to get better before it gets easier. That was the alternative complement pathway. That was the first pathway. Again, the really important thing to realize with this pathway, non-specific, what specifically starts the activation process? That initial C3 floating around is a slight conformational change in that molecule to move that hidden thioester bond to become exposed. That gets the ball rolling. So totally non-specific. Now, the next pathway, the second one to act, is called the lectin pathway. What turns on this pathway? What's the finger that pushes the first domino? In this case, the first domino, the finger that pushes the first domino is actually C3, just C3 changing over time. But with the lectin pathway, it's a lectin that binds to a pathogen surface. And so the question is, what's a lectin? It's not going to be a top hat question, but does anybody know what a lectin is? Does anybody remember and hasn't been super scarred by biochemistry? Anybody brave enough to turn on their mic?
A protein that binds a carbohydrate or can bind.
Absolutely. Don't know who said that, but great work. And I hope you, well, if you were reading from the slide, that's okay too. You were brave enough to turn on your mic, so you get credit either way. A protein that can bind to a carbohydrate. In other words, a protein that can bind to a sugar. Let's make it as simple as possible. That's what's going to bind to the pathogen surface. It's a protein that binds to a carbohydrate on a pathogen. So you're probably going, well, wait a second, Mysick. I mean, holy crap. Lots of organisms have carbohydrates on their surface. Bacteria do, funguses do, even viruses have glycoproteins on their surface. So what makes this, let's call these our pathogens, pathogen, we don't want those. What makes these so different from, you guessed it, the carbohydrates on the surface of all of my cells or the surface of all of your cells? Because we're not a pathogen. We're self, we're us. Once again, we have to think back, and the key part of an immune response is knowing the pathogen, harming the pathogen, and not ourselves. We have to, I like to call it, very biblically speaking, know thy enemy. How do we do that with carbohydrates, with sugars? Well, there are some sugars that are very specific, for example, when we're talking bacteria, to bacteria species that aren't present on mammalian cells at all. They're specific just for bacteria. So we're talking about things like lipoteichoic acid. We find those on gram-positive bacteria, or peptidoglycan. We talked about that the last class, right? This is very specific. We do not have these molecules at all associated with our cells. There's lipopolysaccharide. That's in gram-negative bacteria. Again, this is very specific to bacteria, as well as peptidoglycan, not to mammalian cells. So there's some level of specificity. And it's not just bacteria. For example, for fungal pathogens or funguses in general, yeast, for example, have these high mannose glycoproteins on their surface. You can see all the little green circles are mannose, with this terminal mannose residues at the end, lots and lots and lots of mannoses. Well, that's something we don't normally have, not on our cells, human cells. What we have on human cells, we have lots of glycoproteins, absolutely, but most or many of those have sialic acid residues. That's what factor H binds to, for example, our negative regulator of alternative complement. This is specific for us. We don't see as many sialic acid residues on things like yeast cells. This is where we have some level of specificity. So this is how we start with the lectin pathway of complement to start to know our enemy versus us. When we're talking about the pathogens, their sugars that are specifically going to be recognized by this lectin complement pathway, we have a specific name for that. Pathogen-associated molecular patterns. And you're going to see this word, or what is called PAMPs, over and over and over again. This is the molecule on the surface of that pathogen that our cells are going to recognize and go, aha, you're foreign. You're not supposed to be in this body. That's a PAMP. Now, how do our cells do this? There's our cells, there's self. We have molecules on our cells or produced by our cells that are called pattern recognition receptors, PRRs. And it's these PRRs, they're us, they're the good guys, that can recognize the bad guys, pathogens, through these PAMPs. Now, please realize, and you'll see in a minute, that these pattern recognition receptors is a little bit of a misnomer because not all PRRs are bound receptors that when you think of the word receptor, I always think bound to a cell membrane, but not all PRRs are actually receptors bound to a cell. Some of them are actually secreted by cells and are out and soluble factors. So when we talk about the lectin pathway, and we're talking about the PAMPs, remember, these are the pathogen, these are the bad guys, and what we're going to recognize on the bad guys. We've got things when we're talking about the lectin pathway, like mannose, that are on yeast cells, for example, or another sugar called fructose, or any acylated sugars, such as glucnac or galnac. These types of sugars, much more common on microbial surfaces, which includes pathogen surfaces, than they ever are with human surfaces, right? So this is what we're going to be looking for. These are the components associated with the bad guys, the pathogen, and we're going to be using PRRs, pattern recognition receptors. But like I said, this is a misnomer because the two I'm showing you here are not bound proteins, but they're something called the mannose binding lectin, MBL, or the phycolins. And there's several versions of phycolins. These are soluble proteins that circulate in our blood. And again, are bathing our tissues, not just our blood, but they can get into the extracellular fluids. They're there to recognize the PAMPs. So what are these mannose binding lectins and these phycolins? So there's a picture of a mannose binding lectin. Okay. Again, this is produced as a protein that is produced in our liver, and it consists of two portions. So you're looking at the fully complete final version of mannose binding lectin. But what is made in our livers, you think about it this way, is one of these molecules. Follow this one down here like this, kind of like this crazy lollipop. That's a monomer of the mannose binding lectin. And every monomer is composed of two important parts. The circle down here, that's the carbohydrate recognition domain, or CRD. This is what's going to bind to the sugar on the pathogen surface. And then the rest of the molecule that you see here in kind of off reddish and purple here, that's the collagen-like domain. Now, one of these monomers by itself is useless, right? That's what I have up here. Each individual carbohydrate binding domain, recognition domain, has no affinity for sialic acid, never will, right? Sialic acid is on our cells, but by itself has only very low affinity for sugars that you would find on pathogens like mannose or fructose. Because what needs to happen with the mannose binding lectin is that monomers have to come together. Monomers have to assemble into initially trimers. That you see here, there's a trimer. And then those trimers have to oligomerize so that you get these big bunches. This is what is now seen in our bloodstream. And the most important thing is that these larger oligomers are really important because individually you get crappy binding, period. But when you have a molecule that has oligomerized, a mannose binding lectin that's oligomerized, it's a multimer, right? Multiple, the monomer, 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 blah, 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 blah. Now, with this multimerization, you have increased binding strength. Now you have high avidity, high binding to the sugars that you would find normally on a pathogen like mannose and fructose. There's still absolutely no, even with the monomeric form, multimeric form, there is still no binding to sialic acids. So there is this level of specificity once this whole molecule comes together. The same thing with the phycolins, right? They start off as individual monomers, again, mostly synthesized in our liver, circulate in our blood. They have two component parts of the individual monomer. Think of it again like a little lollipop. You've got a collagen binding like domain, the stem, and then you have the fibrinogen like domain. And those fibrinogen like domains, just an individual domain, no affinity for something like sialic acid or mannose, sialic acid that's on our cells, and very low affinity for acylated sugars that are normally on pathogens or bacteria. But again, these proteins have to come together. They assemble again as trimers, and then the trimers become oligomerized to the large molecule. And it's again, when we have these multimeric proteins, that we have increased binding strength and avidity, especially for the acylated sugars. So on their own, their individual component parts don't bind very well. When they all come together to form these larger complexes, that's when they have their binding strength. That's when they have their specificity. And I like to think of them as bunches of, they look like bouquets of flowers in a way. So in the bloodstream, we've got these mannose binding lectins and these phycolins oligomerized, these bouquets of proteins moving around, but they complex with another protein or two other proteins, what we call the MASPs. MASPs stand for mannose binding lectin associated serine proteases. There's another one we've seen before. There you see in very simple cartoon form. In this case, it could be a phycolin, it could be an MBL. They all assemble or associated with these two MASP proteins, MASP1 and MASP2. Serine proteases, but guess what? They're inactive. They're zymogens. They don't work. Something needs to be done to them. The question is what? We'll start walking through. We won't get very far, but we'll at least start by plonking this mannose binding lectin or the phycolin down because its binding domains have recognized the carbohydrate on the pathogen surface. Remember, now we have a little bit of specificity. When this binding occurs, that pathogen surface, we're going to have changes in conformation. You see, that's a big thing with these complement pathways. Specifically, when this thing lands, let's call it landing on a pathogen surface, there's going to be a conformational change in MASP1. Remember, MASP1 is an inactive serine protease. But that conformational change to MASP1, when it lands, this whole complex lands, causes MASP1 to auto-activate. In other words, causes MASP1 to now be an activated serine protease. And so what's this serine protease going to act on? MASP2. It's going to cleave MASP2 and activate it. And then the next question is, of course, and we'll stop here, but what's MASP2 that has now been cleaved and now has become active? What's it going to start to cleave? We've got this, again, cascading domino effect. I'll stop here. It'll probably take later in the evening. I'll repost this to Canvas because it'll all be recorded. If you have any questions, you can either throw them in the chat now. There's also a discussion board that will be up until our first test. So feel free at any time to put any questions into the discussion board. And I will see all you guys Thursday. Take a look at ImmunoQuest, really cool prizes. And go out and make a snowman. Oh, and I apologize for my cat constantly meowing. He has dementia, and that's what I'm sticking to.